I'm Michel Gomens, the department head, and it's a pleasure to welcome you to this cycle of Simon's lectures. A few weeks ago, we had Simon's lectures in pure mathematics with Boaz Klartag, and so this time we have the privilege of having Ray Goldstein from Cambridge University. So as you know, since many of you come often to these Simon's lectures have been a gift from Jim and Marilyn Simons, and has allowed us to attract some very distinguished people in pure and applied math. And so today we have the pleasure of having Ray Goldstein, the Alan Turing Professor of Complex Physical Systems at Cambridge University. Actually, speaking of Alan Turing, it's a very nice title to have. And for those of you interested, there is a play at the Central Square Theatre until Sunday or Monday. So last few days to see it on Breaking the Code, so by Alan Turing. So Ray's research focuses on biophysics, nonlinear dynamics, complex systems, particularly in the areas of living systems. So he's a pioneer of the physics of life and trying to understand the nonlinear dynamics inside living systems. He's an MIT product, one could say. He's graduated from MIT. His father graduated from MIT. One of his two brothers graduated from MIT. So one of his nephews, I think, graduated from MIT, and probably I'm missing a few. And he has numerous awards. Let me single out a few. He's a fellow of the Royal Society. He received a Bachelor Prize from the International Union of Theoretical and Applied Mechanics, which is given every 4 years in fluid dynamics. He received the Rosalind Franklin Medal and Prize from the Institute of Physics. And just recently, I think a few weeks ago, he received a Doctor Honoris Causa from KU Leuven in Belgium. And so now, if you have seen me at graduation with my furry thing on my gown, he has one too now. So, and for the fun of it, let me mention one last prize. In 2012, he received the Ig Nobel Prize in physics for his work on the shape of a ponytail. And I guess neither of us need to know about that research. So please welcome Ray.
Well, thank you, Michel, very much. I should say that despite, I think it's 11 degrees in our family, we never got a discount. I think I'll take that up with the president. But, well, thank you so much for the invitation to be here. Thank you all for coming. It's great to be back here. When Michel first sent me this invitation, the first thing I did was to look up all the previous speakers, particularly the applied math side. And one of the things I realized was it had been a long time, 20 years basically, since someone had devoted the lectures to really in-depth discussion of biological phenomena. So I decided my purpose here. Oh, it was on. Oh, no. It seems to be on. It was just. No, it goes off. There it is.
Okay.
Oh, there we are. Sorry about that. Should I start over? No, I won't start over. Anyway, it had been 20 years since you had had a lecture devoted to biological phenomena. So my purpose is to fill that lacuna by giving you 3 lectures on one of the most fundamental problems in biology. Now, if I asked you to make a list of the outstanding problems in biology, by this I don't mean will my NIH grant be renewed, but really the unsolved mysteries out there Probably many of you would say that the origin of life is the top unsolved problem, and perhaps the nature of consciousness is second on the list. But very high up there is another question that may not have the public knowledge that the others do, but it's really very important. It has to do with the origin of multicellular life. That is, if you think about the simplest kinds of organisms that first emerged from the primordial soup, they were clearly single-cell organisms, maybe something akin to bacteria. But eventually they evolved to become larger, not only in the sense of having more cells, but in having more cell types and dividing up the processes of life into specialization of cells, like we do. And if you think about life in the primordial soup, many of the interesting and important issues that organisms have to deal with involve things like buoyancy and diffusion and mixing. It's basically a problem in chemical engineering. I think I can say that, unless there's some biologists in the room who might object, but I view it as basically a problem in chemical engineering. And in that sense, it's not surprising that many physical scientists, mathematicians, are drawn to this question of understanding the origins of multicellularity. So that's what I want to talk about today. Now, I should say a few words about philosophy. So I got my undergraduate degrees here in chemistry and physics, and with that I learned to do experiments. But then I went off to do theoretical physics in graduate school. But eventually I came back to my roots and converted myself to do experiments. And one of the important rules in Cambridge, the other Cambridge, is that the names of departments and what goes on inside often have nothing to do with each other. So I may be in an applied math department with string theorists and previously with Stephen Hawking, but we actually have a working cell biology lab. So I'm going to be telling you about essentially a combination of experimental and theoretical approaches to this problem. And I would like to just make a point about the distinction between pure and applied mathematics here, in the sense that if I give you the viewpoint of G.I. Taylor, who was one of the most important fluid dynamicists of the last 100 years, a fantastic scientist, he said it is the discovering of the connection between physical phenomena and describing them by mathematical analysis, rather than the analysis itself, which is interesting. And so, of course, some of you professionally take another point of view. and that's fine. You know, it's a big tent. We can all appreciate the different approaches, but that's my approach. And the other thing I should say is that the great philosopher of science, Yogi Berra, once told us that you can see a lot by looking. And so that's really our philosophy. Now, he also said if you come to a fork in the road, take it. Never mind. Okay, so I want to start with some data, and I think this is the place to begin. So many years ago, Bell and Moores assembled from the literature data on, shown here, on the number of distinct types of cells found in organisms as a function of the number of cells in the organism. So it's a log-log plot over many orders of magnitude showing this. Now there's some agreed upon method of counting here, and this is for organisms all across the animal and vegetable world, from green algae to animals. Now, a human is about 1/10 of a cubic meter in size, and a cell is about 10 microns on the side, so that's 10 to the 14th cells, and we have about 210 distinct cell types. The bacterium is down here at 1,1. Now, if you're a statistical physicist, you might try to find a straight line on this log-log plot, and I'm here to tell you, don't do that. It's very noisy, and I'm not sure what a single exponent will tell you. But it is clear that there's a general trend that as things get larger, they get more complex. And you don't have to look at this graph. You can just appreciate any organization of humans, a university, a corporation. It might have started as two people in their garage writing software or something like that, but eventually it becomes a huge corporation where everything is specialized. So many people, many biologists, many physical scientists have realized that the way to make sense out of this is actually not to consider the whole graph, but to be reductionist, and to look in the lower left-hand corner and ask ourselves, as we move from 1 to 10 to 100 to 1,000 cells, and we suddenly start to see the appearance of several cell types, can we understand that? And I submit that if we can't understand that, we're not going to understand the whole graph. So this is the approach that I want to take here in these 3 lectures. And because this is a very interdisciplinary audience, and this is a very interdisciplinary talk, I will spend a bit more time on the introduction than maybe other lecturers did, because I want you all to be on the same page here. Now, anytime in biology, when you're confronted with these kinds of epic questions, it's natural to look for what are called model organisms. Now, model organisms are organisms which are studied from a variety of perspectives throughout the community: ecological, biochemical, genetic, you name it. And by focusing on that in the whole community, you get a holistic view of what's going on and hopefully a deeper understanding of the phenomena of interest. So if you're interested in plant science, you might focus on this sort of economically irrelevant plant called Arabidopsis, but it's the workhorse organism because you can do genetics with it and all these other things and study phenomena quite broadly. If you're interested in neuroscience, you'll study C. elegans, this little roundworm, which is very important. You can see all the cells and write down exactly where they are, and you know how they're connected. And if you're interested in developmental biology or genetics, then you'll study the fruit fly, Drosophila. So then the question is, for these organisms, what should we look at for these questions that I'm dealing with? And the recent literature from 1700 gives us an answer. So Antony van Leeuwenhoek, who was not the inventor of the microscope, but the perfecter of the microscope in the late 1600s, early 1700s, wrote many letters to the Royal Society about what he saw with this amazing little microscope that is the size of several postage stamps with a fantastic millimetric lens and a little XYZ translation stage where you can hold it up to the light and see amazing things. And one of the things he wrote about in a paper whose title I really want to use someday, what you find in sheep's livers, gnats, and in the excrements of frogs, he reported the discovery of an organism which had a fantastic rolling dynamics about it. It's a beautiful organism that was not named at the time, but about 60 years later, Linnaeus, the namer of everything, in his great book on taxonomy, gave in the last entry in the book the name volvox, from the Latin volvere, to roll or spin, because of this characteristic spinning motion. Now, if you read Latin, which I don't do very well, down here, you'll realize that he described it as rolling around without pause, not P-A-U-S-E, but P-A-W-S, without some means of, some appendages. And he also talks about having children and grandchildren and great-grandchildren, and we'll see that in a moment. But it turns out, actually, the mechanism of motion of this organism involves what are called flagella or cilia, which are very much found throughout our body and play a very important role in biology. So I'm going to spend a few minutes just telling you about this organism because it's so wonderful and perfectly suited to this problem. So here's volvox in the flesh. This is a picture I took many years ago. It's about 800 microns across. And speaking of hair, if you have any hair, typical diameter of a hair is 100 microns, worldwide average. So we're talking about something which is just below a millimeter, 8 hair lengths across. So it's a big transparent object which has cells on the surface of a transparent polymeric goo called the extracellular matrix. They're called somatic cells, and each of them has 2 flagella that allows them to swim through their fluid environment. And then the children or daughter colonies are sequestered on the inside of this sphere and grow up to become the next generation. And they have inside of them what are called germ cells, which are the grandchildren, which will become the children of this one. So it's a beautiful organism with a highly regular structure, and it turns out to be tailor-made for many of these questions. So if you get the BBC in your lab, then you can help them make a nice little visualization of volvox. Here you see it in the flesh. This is real time. It spins around in a beautiful way as it moves. It's photosynthetic. It needs the light. It's not surprising it does this, and I'll have more to say in the future. But it's not just this organism, actually. Clearly, God is a mathematician in the sense that if you look at living organisms now that form what is called a lineage, you'll find that there's a whole collection of organisms that are basically cousins of this one that start with a single-cell organism called Chlamydomonas with 2 flagella. And there are distinct species with 2 to the n cells, where n can be very large, like 9 or 10 or more. All distinct species that may take a plate-like form initially, but eventually are spherical structures that eventually form the proper species volvox. And there can be species of volvox with 50,000 cells. So what you see here is that we go from about 10 microns, so a tenth of a hair, to something that you can easily see with your eyes. So there's a huge dynamic range here of scales where we can investigate the scaling laws that underlie life and the transition from the single-cell organism to the multicellular one. But moreover, there's a very sharp dividing line right here in the sense that all of the species to the left of that line have the feature that every cell in the organism does all the functions of life, which basically means reproduction and everything else. Whereas, by the time you get to volvox, the cells on the outside are, let's call them galley slaves. All they do is swim. They don't reproduce, and all the reproduction is on the inside. So this is what is called germ soma, differentiation, soma short for somatic or sterile cells. So this is one of the most elementary distinctions in biology, and it's the example of climbing that graph in Bell and Moores from 1 cell type to 2. So the natural question is, why did nature put this dividing line here? what's special about this size of organism or the number of cells that suddenly makes it evolutionarily advantageous to have two cell types. So that's kind of the picture you should have in your mind, that we have this Swiss Army knife of species with many different characteristics, but they go through this transition to multicellularity in a very sharp way. Now, this issue of the division of labor has been around for a long time. And when you live in the UK for a while, you realize that there's a dynasty of the Darwins and the dynasty of the Braggs, and there's the dynasty of the Huxleys. And Julian Huxley, at the age of something like 32, wrote this amazing book in 1912, exposing all the issues around the individual in the animal kingdom. Where does this division of labor come from? What does it mean? How is it selected from the point of view of nature? And he has this beautiful drawing of Volvox. This indicates we've all lost the ability to look through the microscope and actually draw things, but people then were amazing. But there's this quote, which is fantastic. It was thus the other method of aggregation of similar units and subsequent division of labor among them that opened to life the full resources of what he called the second grade of individuality. In some colony, like Volvox, there once lay hidden the secret of the body and the mind of man. So that, I wrap myself in that and say that's justification for studying this kind of problem. Okay, so the last thing I want to say introduction-wise is some comments about the role of fluid mechanics in biology. Once I was visiting a major North American university a bit to the north of here. I won't say which one it was. But I was met there by an astrophysicist who asked me what I worked on. And I said, well, I actually work on a lot of biological fluid dynamics. And he looked at me and he said, is fluid mechanics important for biology? And I wasn't quite sure what to say, except to remind him that he had a heart. but I thought about it for a while, and then I realized, actually, the most interesting answer to this astrophysicist was actually Leonardo. So if you remember the Vitruvian Man, one of his most iconic drawings from the late 1400s, it's a celebration of the proportions of the human body, the fact that your wingspan is pretty much the same as your height, and there's a beautiful illustration of bilateral symmetry that we all have to a very good approximation. But as you may know, symmetry is only skin deep, because if you actually look under the skin, you'll discover that your heart, most of the time, most people have their heart on the left-hand side of their body. That's not always true. Some people have the heart on the right-hand side of the body. And in fact, their entire body plan flipped. And those who have, okay, I should be less excitable. Whoa, sorry, let's go back here. Whole body plan flipped. Let's see if that works. Yes, okay, sorry about that. It was the unraveling of the mechanism by which that left-right symmetry breaking happens that is one of the most interesting stories in biology in the last 20 years or so. So we all have, roughly speaking, 3 broken symmetries: top, bottom, front, back, and left, right. And in early embryo development, it is the left-right symmetry which is the last one to break. And what was understood, starting around the year 2000 or so, is illustrated here. So on the left, you see a micrograph of a mouse-to-be, a mouse embryo. And in that little square is an object known as Henson's node. It's more generally known as the left-right organizer. And inside this chamber, which is kind of pancake-shaped with a membrane on the top, there's a whole collection of these hair-like appendages called cilia that rotate around in a very particular fashion. And through a process that's still a bit mysterious, despite all the time that's passed, it sets up a differentiation of chemical messengers between left and right that ultimately determines where the heart goes. And we know this is true because the experimentalists who discovered this peeled back the lid, stirred the fluid the other way, and the mouse ended up with its heart on the other side. And if that doesn't illustrate that cilia are important and fluid mechanics is important, I'm not sure what would. So that's my answer to the astrophysicist. Okay, so in the last 20 years or so, it's become possible to use these green algae as I say, a kind of Swiss Army knife to study lots of problems in biological fluid mechanics as well. So you can study the issue of the development of multicellularity. You can discover accidentally things like what you see here called a hydrodynamic bound state, where these spinning organisms near an upper surface through the surface-mediated hydrodynamics actually attract each other and spin around each other, kind of like a bound state in quantum mechanics. Because they're large, I mean, half a millimeter is large in my world, you can actually measure very accurately the fluid flow around them and try to relate the measured fluid flow to the action of these thousands of flagella on the surface. As you see here, you can study things like the distribution of tracers in this system kicked around by these swimming microorganisms as a kind of pseudo-Brownian dynamics. You can study, as you see here for Chlamydomonas, the synchronization between these two cilia, which will be the subject of one of my talks, how they manage to steer toward the light. They're all photosynthetic and phototactic. They swim toward the light. They don't have a central nervous system. How do they do that? And more recently, it turns out they're models for understanding aspects of tissue folding. So it's really quite an interesting set of organisms. So what I'm going to talk about in my 3 talks is shown here. The first one today is going to be about the synchronization of the cilia or flagella, interchangeable words, on these organisms, and how, maybe through a little bit of a mystery story, I'll show you the back and forth of the scientific method as a way of ultimately getting an answer to how do they synchronize, because wherever we see them, they are synchronized. The second talk will be about this tissue folding problem, where we'll look at how basically a sphere can turn itself inside out. That's a well-known, famous problem in pure mathematics. Biology manages to do it its own way, and we'll talk about that. And the third talk will be about how these organisms manage to find the light. They don't have a central nervous system. There are 1,000 cells in Volvox. They don't really talk to each other that much, yet it manages to go there. How does it do it? How does it make a decision without a brain? Okay, so on the subject of synchronization, there is a long history of study of this problem, and you can see in many different biological contexts examples of this. So the one at the top is nearby swimming sperm cells, as first studied by Lord Rothschild in England many years ago, where as they swim near each other, they nest together so closely you can barely tell them apart. Let's call that in-phase synchronization. When I tell you about Chlamydomonas, we'll see it has a breaststroke motion, and like a good breaststroker, they're very, very much in synchrony, and we'd like to understand that. But even if you look at bacteria, which swim a very different way, they have helical flagella that are turned by rotary motors in the membrane. They can bundle together and unbundle in a way that is ultimately about synchronization. And finally, in various ciliated tissues in you, the back of your brain, your trachea, reproductive system, you will find carpets of cilia that are not perfectly in phase. They actually have metachronal waves, as they're called, that are like Mexican waves in a stadium. So there are all these different ways in which these appendages can synchronize, and we'd like to understand how. So I'm getting near the end of the introductory biology. I know it's a lot, perhaps for a mostly mathematical audience, but I want you to appreciate what we're trying to do here, which is somehow solve a physicsy problem with a clear understanding of the biology. So here's Chlamydomonas, that single-cell organism, and if you take a very microscopic look at the cilium or flagellum, you'll discover that it has a microstructure that's very characteristic. It's made of polymerized proteins that are kind of like soda straws, and there are 9 pairs of these things called doublets around the periphery of this half micron or so cross-section, and 2 in the middle, and that so-called 9+2 arrangement is one of the most highly conserved structures in biology. It's exactly the same in you as it is in Chlamydomonas, but Chlamydomonas appeared on Earth the better part of 1 billion years ago. So that conservation is a good indication of how important it is to life. And the way that this object moves is that there are proteins called dyneins which attach to them, basically the back attached to one doublet and the front walking along the other. And if you slide filaments past each other by that motion, they bend if they're clamped at the base. And so there are the waves of bending that allow the entire motion to occur through the translation of these motor proteins. Now, I was going to show you a lecture demonstration, but I discovered that the floor in this room is sufficiently out of horizontal that it doesn't work. So instead, I have a video substitute, which is to remind you of something you may have heard about. In 1665, Christian Huygens, who invented the pendulum clock, discovered a very interesting phenomenon that 2 pendulum clocks on a wall, swinging their pendula back and forth, will actually synchronize. And there's a very nice video demonstration if you can't run your actual demonstration. I have metronomes over there, and afterwards you're invited to play with them. So here you have 5 metronomes, and even if you don't hear them, it doesn't matter. 5 metronomes, all at the same frequency, sitting on an arrangement in which there are 2 soda cans, empty, and a lightweight support that allows some motion back and forth. And even though they were started with random phases, after about a minute, you will discover that they all synchronize very nicely. Now, Huygens' pendulum clock synchronized out of phase like this, That's because there's no lateral motion of the wall. There's just flexing. But this thing can move. And after a fashion, a little bit more, they all synchronize. So this is one way to get synchrony out of oscillators. And a pendulum goes back and forth through its cycle. A flagellum goes back and forth through its cycle. It's an oscillator. So the question I want to answer, I hope, is what's the equivalent of the wall of Huygens or the soda cans and the Planck? What allows synchrony? Is it some flexibility in the system? Is it the fluid between the flagella? This has been an outstanding question for a long, long time. And the community together has, I think we can fairly say, finally answered this. Okay, so just a couple of mathematical tidbits to get us in the right frame of mind. First thing to say is that we're working on a very small scale here, things that are seen under the microscope. And if you think about the Navier-Stokes equations as the fundamental governing fluid equations in the problem, as I've written up here, left-hand side represents acceleration, rho, the density of the fluid, du dt, the time rate of change of the velocity, an advective term representing the fact that if there are gradients in velocity, they get carried into your point of view, your field of view, by the fluid velocity. And on the right-hand side, you have the gradients of pressure, a viscous dissipation term with mu as the viscosity, and some body force that may exist. If you just do a scaling analysis and ask yourself, well, if there's some characteristic velocity u, some characteristic length l, how do these different terms appear? Well, the left-hand side would basically clearly look like rho u squared over length, because the gradient is 1 over length, whereas the dissipation term is mu u over l squared. So the ratio of these two is what we call the Reynolds number, advection over dissipation, and it looks like ul over nu, where nu is the viscosity divided by the density, called the kinematic viscosity. And in the units I want to use, the kinematic viscosity is 1,000,000 microns squared per second. It's a diffusion constant. Okay, now if you're in the Stokes regime, as we call it, where inertia is irrelevant, the Reynolds number is essentially 0, which is the case for organisms of the size we're talking about, maybe 10 to 1,000 microns, swimming 10 to 100 microns per second, the Reynolds number is 10 to the minus 4 to 10 to the minus 1 or 2. So you can just neglect it. There's no inertia. If you stop swimming, you stop moving. And in that limit, if you have a point force, which is the only fluid mechanics problem I need to teach you about today, a delta function at the origin, then the characteristic flow around that, which is called a Stokeslet, looks like this. Falls off like 1 over r, and there's this anisotropic tensor that multiplies your force. And here are the streamlines of a Stokeslet pointing down. And here's an experimental measurement of the velocity field around Volvox. It's denser than the fluid around it, and so it's a point force acting on the fluid, looked at from a distance, and the two match beautifully. Now, you can also ask about things like the transport of some chemical messenger in the system. After all, the organisms may deal with carbon dioxide, dissolved ions, oxygen, et cetera. So if you have a concentration C of such chemical species, it will be advected and diffuse around with an advective term and a diffusion term that scale like this. And the ratio of those two terms, advection over diffusion, looks just like the Reynolds number, but now you have the molecular diffusivity of the chemical species. Now, the typical diffusivity of a chemical species in the same units I was using is not 1,000,000, but 1,000 microns squared per second. So this Peclet number is already automatically 3 orders of magnitude larger. So for an organism like Chlamydomonas, the Peclet number is barely 1, if you're lucky. So this is a diffusion-dominated world. But this organism, so much larger, with the collective behavior of all of these flagella, swims so much faster, it's actually in a large Peclet number limit. So actually, the transition of life from unicellular to multicellular, at least in this lineage, is a transition from a diffusion-dominated world to an advection-dominated world. And that makes for all sorts of interesting consequences, which I will hopefully have a chance to get to. Okay, so as I said, we're going to be doing some experiments here. And the way we do this is basically to use techniques that are borrowed from the world of in vitro fertilization and electrophysiology. So we use micropipettes, which are glass, I've learned the British pronunciation, capillaries, which are necked down to tiny sizes so that you can hold a single organism like that or larger organisms. And we have all the infrastructure needed to do this. We have chambers in which to grow these things on a day-night cycle. We have all the infrastructure to do the biochemistry. And most importantly, we have the ability to hold these organisms for interrogation under the microscope. And the most important innovation that we did in all of this was seen right here, this glass capillary, which actually holds the microorganism for interrogation here, is on a rotation stage. So if we grab an organism and it happens to be like this, we can rotate it. So its flagella are in the plane of motion, and you can actually watch them quantitatively. Okay, so here's an old but sentimental video just to show what you see when you look at the edge of Volvox. So remember, the organism is huge on the scale of the individual somatic cells here. This has been image processed for your viewing enjoyment. And what you watch here are the galloping motions of these two flagella. And you'll be excused if you think, well, sometimes it appears like they synchronize and then they don't, and what's actually happening here? How do we quantify what's going on? But it's pretty clear there are some interesting dynamics going on, even at the level of those two. So the first person to try to understand this was G.I. Taylor in 1951, who wrote a very beautiful paper where he abstracted the whole problem of cilia flagella to basically say, just consider two basically undulating carpets in a viscous environment. Actually, first let's consider one, and then we'll put two next to each other, and solve the fluid mechanics of that problem. Now, in the Stokesian regime, the fluid mechanical equations themselves are perfectly linear. And if you express the velocity in terms of an underlying stream function, psi, the problem you have to solve is basically the biharmonic equation. And you might say, well, that's a perfectly linear equation, so what's the big deal? Well, the big deal is that if, for instance, you go into a moving coordinate system with one of these moving carpets, the boundary conditions that you have to satisfy are that the fluid moves with the material points. And that is a boundary condition on the stream function that says, for instance, for the x component, I better have zero velocity in that moving frame evaluated on the sheet, and the sheet may have some small amplitude in front of it, but that means that I'm solving a problem in a half space for which I do not know the Green's function. So what is done is what is known as boundary perturbation theory. I systematically, order by order, in the amplitude of the perturbation, map the system back to a flat domain where I do know the Green's function and I can write down the solution. But this creates a very complicated calculation, but Taylor carried this through. He was able to show a single sheet actually does swim, even though there's no inertia at all. But more importantly, he put two sheets together, solved the problem in the three domains, and asked himself how much energy dissipation is there in the problem. And the rate of energy dissipation is the volume integral of the square of the gradients of velocity. So it's pretty clear that if the sheets are nested together, they're smaller gradients of velocity than if they're 180 degrees out of phase. So if you calculate the ratio of the energy dissipation in phase and out of phase as a function of the spacing between the sheets, you discover when they're far apart, it's 1 because they don't talk to each other. And as you move them closer, there's a dramatic reduction of the dissipation for the in-phase state. So Taylor's hypothesis is that what we see in nature, the fact that these are synchronized, is nature trying to minimize dissipation. It sounds very plausible. I mean, most of us do that in life. But I want to remind you that minimizing dissipation is not in general a principle from which you can deduce dynamical behavior. All he did was place it at different positions and say, by comparing them, I deduced this. My colleague Eric Loga and Elfrink, some years later, actually sat down and added some elasticity to the problem and discovered that you can actually get a dynamical evolution to the minimum dissipation state. So that's the start of an understanding that you have to have not only hydrodynamic interactions between these objects, but some flexibility in the system.
You're saying it, what is the horizontal axis of this graph?
This, the spacing scaled by the wave vector of the undulation. So as you bring them further apart, they don't talk to each other, so the ratio is independent of the phase. But as you get closer together, you can drop the dissipation by a factor of 3 by being in phase relative to out of phase. Okay, so now let's talk about historically what people knew about the dynamics of the model organism Chlamydomonas, because it's perhaps the most interesting one. So Ruffer and Nulch in Germany many years ago did the first pioneering experiments on this problem. They did micropipette holding of the organism, and at the time, 1987, there were no digital high-speed cameras, it was film, and they analyzed frame by frame the images on the film by hand, drawing the waveforms, and from that deduced what you see here. So this is a heroic experiment. So 85% of the time when they fished one of these Chlamydomonas out of solution and held it and characterized it over the course of half a second, if they looked at the frequency of the two flagella, one is called cis, one is called trans, relative to the photosensor that they had, they were perfectly in sync. So it's breaststroke motion, slight wiggles in the frequency, but basically synchrony. Then about 10% of the time in this period of time, they would see one of the flagella suddenly have a higher frequency and come slightly out of sync with the other, and then return. Transient asynchrony. But about 5% of the time, the two flagella didn't seem to coordinate with each other at all. They had observably different frequencies, perhaps as much as 30% different. Now, as I look around this room, I can try to make an analogy. Some of you are more coordinated than others. Right? And so some of you walk gracefully, and some of you maybe not. And so the assumption that Ruffer and Milch did was that these are distinct subpopulations of cells. It's a natural assumption. There's variability in life, especially at the single cell level. So the question is this really true? Or is what we're seeing here natural variations or something else? So to study this problem, we use the technology that I showed you, and many years ago started to try to quantify this problem using the language of nonlinear dynamics, the language of oscillators. So this is a highly image-processed remnant, it's soon to be a video, of an image of Chlamydomonas. Here's where the cell body was. Here are the two flagella, and here's what's left of the micropipette. And we're going to basically imagine that each of these flagella executes some motion, a stroke, let's call it, S1 and S2, with some amplitude and the cosine of some time-dependent angle. And that angle would be a way of talking about where they are in their cycle. And if all there was were two independent oscillators at different frequencies, then trivially the time rate of change of those angles would just be omega 1 or omega 2, but there'll be more. And if I just divide by 2 pi, I could read off the frequency difference by apparently finding a straight line behavior in time, and let's see what happens. Okay, so here's a video over the course of several seconds. This thing beats at 55 cycles per second, so this is a high-speed image at about 2,000 frames a second. And as it goes along, you see this characteristic synchronized breaststroke motion. But just wait. At some point, yep, you see, it went out of sync, and then it came back. And so that was a perfect example of the 10% of the time observation of Ruffer and Milch. Now, they could only do their experiments over half a second or so because it was just so tedious. But let's see if we can do a bit better. So the first thing is, how are we going to analyze this? And at the time we first did these experiments, we essentially made a Poincaré section of the problem. So you erect two little interrogation windows on either side of the cell, and each time a flagellum comes through that window, you see a big peak in the pixel intensity, and you say, it's going through a cycle. And from that, you can just linearly interpolate to get the difference in angle as a function of time here over about 4 seconds. And what you see are two things. First, it's a noisy signal that transitions over a fraction of a second from one value to one that's one integer different. So that's what we call a phase slip for oscillators. But it's noisy. And you can do a simple calculation. You can ask yourself, if I had a heat bath, thermal equilibrium problem at a temperature T, and I had an elastic filament with the stiffness of a flagellum, how much noise would I get in this quantity? And it's 3 orders of magnitude below this. So this is not thermal noise. This is actually intrinsic biochemical noise in the beating. It's stochasticity, but it's not thermodynamic, fundamentally. Okay, and so the thing is, though, that our high-speed camera has a lot of memory on it, and we're not restricted to 4 seconds. We can look at, say, a minute. And so for a single cell, this is what you see for that quantity delta. It's all over the place. And you see, first of all, there are periods of noisy synchrony, just like I showed you. There are periods of phase slips, up or down. And there are periods of drift where you basically get straight line motion. So it's not that it's distinct subpopulations of cells. Each cell, stochastically, over time, is visiting these 3 behaviors. And therefore, it is exactly like in statistical physics, the ergodic hypothesis. I can take a snapshot of everybody in this room and take an average, or I could follow Roger over time and just do a time average, and I'll get the same answer. It's not obvious at all that this is what should hold for a biological system. But it does, because when you look at the fraction of time spent here, it's 85, 10, 5, within experimental error, exactly what Ruffer and Milch saw. So that's the first thing. The second thing is, can we have a model to try to do something with this data? Because right now it's just pictures. And so here I'm going to make a slight aside on modeling to say that if you make a measurement of the flow field around these organisms, Chlamydomonas doing this. This was a heroic experiment done by my then PhD student, Knut Drescher. This is what you get. So this is obtained by looking at thousands of trajectories of cells as they swim in and out of the focal plane with microspheres to track the flow, and from which, by reorganizing the flow by the direction, you can consolidate everything and get the average flow field of this organism. And these swirls on the two sides are from the flagella doing this. And what you see is that there's a stagnation point in front, the two swirls, the flow comes in from here and in from here. And this is a very characteristic so-called stresslet flow that's a forced dipole to two Stokeslets of opposite direction. But the amazing thing is that there's a simple model for this in which you think of the cell body as acting as a point force on the fluid, and roughly speaking, sort of the elbow of each of the two flagella as a point force acting back, such that the whole thing is force-free. And that three Stokeslet model is a pretty good representation of the entire flow field, which is ex post facto justification for models of flagella which treat them as little spheres moving around on orbits. And I'll tell you a little about that model because it's a good way of understanding what goes on. So this model was fiendishly clever by Niedermeyer, Eckhart, and Lenz. They said, let's just forget about all the complexity of flagella and talk about a sphere moving on an orbit with some force pushing it around tangentially, represents the dynamics of the flagellum. Let's say two of them separated by some distance L. And each of those little spheres is held by a spring so that there's a penalty in energy to move away from some preferred orbit radius R0. So as these things are whizzing about, they're going to be pushing fluid, pushing on the other one, just like those pendula were doing. And this will make them change their dynamics, and maybe they will synchronize. So mathematically, in the azimuthal direction, there's some drag coefficient times the velocity r phi dot in the azimuthal direction minus the component of velocity due to the other sphere, and that's set equal to the force that's driving them around. And in the radial direction, you have the change of the radius with some drag coefficient, the equivalent velocity contribution, and some spring with a rest length r0. Now, as in many problems in physics, one expects in this one there's a separation of timescales, that the spring is very stiff, so the radial motion is very fast relative to the azimuthal motion. So there's a slaving dynamics you can do here that results in essentially saying having an algebraic relationship for the radius of, say, particle 1 relative to the preferred radius, given in terms of the angular velocity of particle 2. And likewise, there's a coupled dynamics of the angular velocities. So if you go to the limit where these are weakly coupled, you can solve this algebraic problem precisely. And what you see is, first of all, that this velocity field from one of the spheres on the other is a Stokeslet flow, because it's just a sphere moving around, falls off like 1 over the distance. And when all the dust settles, you end up with the dynamics that I show you at the top here. So our quantity delta, which is this phase difference divided by 2 pi, has delta dot involves the intrinsic frequency mismatch, which we argued was obvious. And then you end up with a function that represents the coupling between the two oscillators, which is periodic in delta, as it must be, because these are oscillators. So this equation by itself is called the Adler equation. It's a simple nonlinear ODE. Adler was the inventor of the television remote control. So we owe many things to him. But what I told you was that this was a noisy dynamics. And so in the simplest picture of noisy dynamics, we would add to this some stochastic forcing with, say, zero mean, delta function correlated in time that would represent some kind of effective temperature, like in statistical physics. So now we have a Langevin equation for a noisy Adler equation. And the first question is, how does this work? And it's actually very simple and very interesting. So imagine I have two spheres going around in these orbits. And let's suppose this one is lagging behind this one. Well, the fluid flow from this pushes this way, which will push this one out. But because the force that's pushing it around is a constant force, doesn't change in magnitude, if I move to a larger radius, I must slow down my angular velocity to have the same force. So this one lags behind, it'll push this one out, and this will slow down, and therefore this will catch up. And if this one happened to be leading, it will push this one in, which will make this one speed up. And the same holds in the opposite way. So it's a natural way to get synchrony, and it involves two things. The first is hydrodynamic interactions and what you might call orbital compliance, the fact that the orbit is flexible, just like I told you earlier. However, there's a point to say here, which is, as Raymond Goldstein and others have argued, you can actually replace some of this by basically talking about a phase-dependent forcing without the orbital compliance. So either of those mechanisms will work. So now you have a mathematical problem. I give you a data set, and I say, tell me the parameters of the problem. So I'd like to know the frequency difference, I'd like to know the coupling, epsilon, and I'd like to know this effective temperature. How am I supposed to do that? And there are three important methods of mathematical physics that I know of. One of them is adding 0 to both sides of an equation. The second one is multiplying by 1. And the third is mapping it onto a previously solved problem. And so this is how you do it using the third method. So you look at this dynamics and you realize that this is actually a noisy gradient flow. That is to say that delta dot is the right-hand side is the same as saying there's a potential v of delta whose gradient is driving the deterministic terms. And that v of delta is a tilted washboard potential. It's a term linear in delta and a sine wave. And so it looks something like this. So what you realize is that the dynamics we're seeing is equivalent to a noisy Brownian particle moving on a tilted washboard landscape. And so we can interpret what's going on three ways. We can say, if I hop to the right or to the left, there'll be a difference in probabilities that will be given by the Boltzmann factor of hopping over that barrier. That's one relationship I can measure between the quantities of interest. And the second thing is to say, actually, that when I'm in one of these synchronized states, I have a noisy synchrony, and the autocorrelation function of the noise in that state is basically an exponential that dies off with a characteristic timescale, which is set by the coupling strength and the difference in intrinsic frequency. So even though you're looking at a synchronized state, you can measure the intrinsic frequency difference. Okay, so terabytes of data later, you can actually make a probability distribution plot of the number of occurrences of this coupling constant in units of the mean frequency. And amazingly, it clusters very close to the estimate you would make based on this sphere coupling model, that the coupling is related to the natural frequencies of the left and right flagella, and the viscosity of the fluid, the size of your sphere, the length cubed of your filament, because that's the spring constant of a filament, and the bending stiffness, all of which we know. So it really seems to be consistent with the idea that it's hydrodynamic coupling. But more importantly, when you measure in a scatter plot the intrinsic frequency difference and the effective temperature, you see this huge gap between the two behaviors. This is the synchronized state, and this is the free-running drifting state where the two frequencies are very different, with nothing much in between. So this makes it look like Chlamydomonas has two gears. Basically has a low gear where the frequency difference is small enough that the coupling synchronizes the flagella. And then there's a high gear where the coupling strength is insufficient. The frequency difference is large enough that that washboard has no minima, and you go running along and drifting. And so the system is stochastically switching between these two states on top of stochastically oscillating around the synchron. That's the bending modulus of the flagellum. Yeah. Okay, so if it's true, then basically we have this funny expectation that every once in a while Chlamydomonas is desynchronizing its two flagella. So one is moving faster than the other. Now, I don't know about you, but if my right foot were walking faster than my left foot, I would turn. Of course, I'd then fall, but I would turn. And so it stands to reason that if I do this stochastically, I should execute some kind of random walk. So here's a way to see what's going on. You take a cuvette full of Chlamydomonas, you centrifuge all the cells to the bottom, and then you let them refill the container and see if this random motion is really true. And if you make a plot of the concentration as a function of height, you see something like this, and you can measure the flux of cells across any fiducial line and the concentration gradient there, and you can plot flux versus gradient. And if you get a straight line, that's called Fick's law. And from that, you get a diffusion constant, which in some units is 10 to the minus 2 centimeters squared per second, which isn't meaningful until you think, well, dimensionally, a diffusion constant of a motion in which I swim at speed u, and every tau I turn on average must be u squared tau. And I know u is about 100 microns per second, so somewhere there's a 10-second time constant in this problem. So here's how you get at that. You build a three-dimensional tracking system that consists of a dual view of cameras, one of which gives you x, y, and the other gives you y, z, so that you can construct x, y, z trajectories. And you have to make sure it doesn't have thermal convection or anything like that. And when you do that, you can actually track the swimming of this organism with great precision. And this is what you see. Here's a typical trajectory of the swimmer, color-coded by the angular turning velocity of the tangent vector. And when you look at that angular velocity, you see sharp peaks, which represent turns. And you can threshold, I keep pressing that, sorry. You can threshold that, and then you can look at the statistics of those turns. And what you see is that the probability distribution of turning rates is peaked up around 1 radian per second. But you can look at the PDF of the time spent in those turns. And what you see in blue is shown here. So these are free-swimming cells, just moving around, the time spent in the turn. But you can also ask, for the ones we held, how long did they spend in these drifts, the states where the two flagella were observably different? And it coincides perfectly. So every time we saw them go out of sync while holding them, it was the same as when they were swimming freely. And the PDF of the free flight time between turns is an exponential with an 11-second timescale. So basically, that proves it, that Chlamydomonas is doing some kind of run and turn motion stochastically. Now, if you really want to understand this, there should be some dependence on the flagellar properties. And one way you can get at this is by using a process that is known as autotomy, self-cutting, where you can actually coerce one way or the other the flagella to be lost by the organism, and they will regrow it in about 90 minutes. Now, 90 minutes is a huge time. We can do one of these experiments in a few minutes. So we can deflagellate them, and as the flagella grow back, we can measure what's going on. And what you see, as you see there, is the length as a function of time is this, matches pretty well this model for the reassembly of the flagella, which I won't go into, but it's called the balance point model. But the main point is that there, it takes about 90 minutes, it goes pretty much back to where it was. And if you then look at the quantity delta as a function of time since deflagellation, when they're very short, 10 minutes, 20 minutes later, you see no synchrony, just a straight line motion. And as you get longer and longer, you get these longer plateaus representing stronger and stronger synchrony. And the theory says if you look at the coupling strength to the 1/3 divided by the frequency to the 2/3 as a function of length, it should be linear. And it is. So that kind of shows that the elastohydrodynamic coupling idea makes sense. But nature provides us with more than just the wild type organism, the one you find in the River Cam or online from a culture collection. So the wild type shows this in-phase synchrony that I've been telling you about. But there are various mutants around, one of which is called PTX1. It's a mutant that doesn't do phototaxis very well. And if this is basically the breaststroke, then this is the crawl or the freestyle. So it turns out its synchrony is antiphase. And we'll talk about what is going on with this organism, but that's a good model to study to try to understand exactly what's going on with synchrony. And when you study as a function of time this quantity delta, you discover that it transitions stochastically between in-phase and antiphase motion. So delta is jumping up and down in half integral values, and there's an associated frequency shift going on there. And if you actually look at some simple way of quantifying this, you look at the angle of the two flagella as a function of each other. In the in-phase state, they're going like this, so they go up the diagonal. In the antiphase state, for a single cell, it's just the opposite. And if you average it, it's really clearly antiphase, precisely 90 degrees rotated. So we have this idea that hydrodynamic interactions are important. We have this idea that something else is going on to create antiphase state. And it turns out what PTX1 is missing are elastic connections between the bases of the flagella. So when PTX1 is doing its thing, there's no elastic coupling, but the wild type actually has an elastic coupling, which I'm now telling you about. And somehow removing that coupling changes the nature of the problem. And if you go back to this bead spring model of Niedermeyer, Eckhart, and Lenz, what you discover is that when the flagella are going like this, the natural hydrodynamic coupling is antiphase. So this is actually the simpler problem, even though it's the mutant. And what's going on here seems to be associated with internal coupling. So finally, you think, okay, I know the experiment to do. We'll call it the ultimate experiment. You take two cells, somatic cells of Volvox or Chlamydomonas, you pull off one flagellum off of each. Now you have one flagellum on this one flagellum on this one. There is no elastic coupling between them at all. And there can only be hydrodynamic coupling. And you see what happens. And it's beautiful. What you discover is that as you change the spacing between the cells, you will find, as you go to increasing separation, that basically there's stronger and stronger synchrony with essentially this behavior. And now you can ask yourself, essentially, what is the strength of the coupling that you deduce as you change the spacing. So here you see it precisely. They're close together, very strong coupling. As you move further and further apart, they get further and further decoupled. And they're beating in the same direction, so it's like the Niedermeyer-Eckhart lens model. And if you rotate one of them, so now one is beating one way and one is beating the other way, then you discover antiphase synchrony. So we have now shown that a sufficient condition for hydrodynamic synchronization is, a sufficient condition for synchronization is hydrodynamic coupling, and it will lead to in-phase motion for this kind of behavior and antiphase motion for this kind of behavior. But what's happening in the real system shows the importance of distinction between necessary and sufficient. So the real mechanism in the cells is actually these elastic couplings between the flagella. So the flagella are anchored in what are called basal bodies inside the organism, and there are filamentary couplings between them, which are like the wall of Huygens. And so you can actually look at mutants in what's called VFL that actually have more than two flagella. And then the orientation of these basal bodies is somewhat random. So here you see two next to each other that are beating in the same direction, and they're synchronized in phase, and two here beating in the opposite direction, and they're antiphase synchronized. So finally, what we can realize is that it is a combination of hydrodynamic and elastic couplings. And one way to see that is to take a quadriflagellate. So there are algae that have 4 or 8 flagella, and you can watch their dynamics over time and literally go in and hold one. And if the hydrodynamic interaction was so important between these, the act of holding that should have short-circuited the hydrodynamics and changed the dynamics, and it doesn't. The beating dynamics is pretty much unchanged completely. So you have to be careful about the idea that just because the magnitude of some quantity you measure agrees with the magnitude estimated by theory, that you therefore understand what's going on. The sign matters too. Okay, so the final thing to say is actually if you look very carefully at all the different organisms that have 2, 4, 8, even 16 flagella, you discover that the biologists have long ago mapped out the dynamics or the interconnections between these basal structures. And they're very intricate filamentary connections. And associated with each kind of symmetry of this interconnections, there's a different symmetry of the gait. So you can find examples of horse-like gaits, galloping and trotting and pronking and all of these things in these organisms driven by these filamentary couplings. Okay, so I'm going to stop there and basically say we learned an important lesson here, which is that when you look at a phenomenon and you think you understand it, as G.I. Taylor, I'm not trying to take away his greatness, did by saying, I look at hydrodynamic coupling, I think, well, that's what's going on. And the whole community goes, yes, it must be hydrodynamic coupling. And we all go down this path. You have to step back at some point and use the power of the biological systems, the mutants, all of these things to try to actually understand. And then you discover actually Huygens was right. Thank you.
Any questions?
It's on. Is it on? Oh, yeah, there we go.
It's on.
You just showed that video with the close cilia beautifully coupled, and then as you move them further apart, they became completely uncoupled. The coupled video, it looked like that was just a video of a single cycle. Did they stay, like, have you observed that over periods of time? Do they stay nicely coupled that way?
They do. They stay for very long periods of time, many hundreds or thousands of cycles. Not forever. Eventually, there's the stochasticity can hop them around to other states, and the cells are not perfectly periodic over time. So eventually, just like Chlamydomonas, eventually there'll be some internal biochemical change. But they can be stable for thousands of beats.
Oh, wow. Thank you.
Thank you. Great lecture. I have in the wild, actually, are those organisms living together? Because you seem to study them in isolation. And so I was wondering if there was hydrodynamic coupling across specimens. For example, when you did this centrifuging thing where they're all together and they're all coming up at the same time, it could be due to interactions or is it just totally idiosyncratic?
So the volume fractions that we achieve in the centrifugation are still way below the levels at which you would start to see serious hydrodynamic interactions between the swimmers. Now, Chlamydomonas is an example of what's called a polar stresslet. That is to say, the flagella are forward of the body. So the force dipole of interest is pushing back from the flagellum and forward from the body. Bacteria are just the opposite. Their flagella are behind, and the force is going that way, and the body force is going this way. And it turns out in the theory of concentrated suspensions, the polar stresslets tend not to organize much in any collective way, but the pusher stresslets form a kind of turbulent state, which is characterized by long-range coherence, vortices, jets, all sorts of things. So in some ways, Chlamydomonas is kind of immune from any large-scale coordinated behavior until you get to extremely high concentrations, but we're nowhere near that. That's not to say that there can't be collective behaviors, and in some ways these metachronal waves on the surface of Volvox are examples of some sort of collective behavior, but it's a somewhat weaker coupling than what would be necessary for that.
So in your experiments, you're holding this poor guy still. Does that affect what its behavior might?
So because there is therefore a net force on the object, the long distance flow field will be that of a Stokelet. And that's not the same as a free swimming one, which has, with the exception of gravitational effects or something like that, doesn't have any force on it. And so the long distance flow is that of a stresslet, which falls off as a faster power of R. But for these near field kinds of things, it doesn't matter much. And you can compare the two situations and it doesn't seem to affect it.
And it's too simple an organism to know that it's stuck.
I wouldn't say that. I mean, some of these organisms, for instance, are not these, but they're examples of marine organisms like dinoflagellates, which if you hold them on a pipette like this, they emit bursts of light. So, I mean, probably you would too if I did this to you, but
worse.
You know, they clearly know that their force is being acted on them, and you can get effects, which is why, as you'll see in tomorrow's talk, if you come, it's necessary to try to develop methods to hold them as gently as possible.
Hi, thank you very much for the talk. I have a couple of questions. One is, so this out of phase that you observe would be the tumbling. If one would be to model this organism, it would model it as a run and tumble particles, and then the tumble would be the reorganization, randomization of the direction, which would allow the organism to explore space. Is that true?
So let me choose a slightly different word. So in bacteria, we have run and tumble, where the flagella are synchronized and bundled together into a coherent helix, and you have straight line motion, and then one or more motors reverses, and you have what I call a bad hair day, and the whole thing tumbles, but it tumbles for some period of time randomly. This is run and turn. So you swim in a straight line for a while, and then you just gracefully turn, and then you resynchronize and move off. So on a large enough time and length scale, they look pretty much the same, but we call it run and turn for that reason. Now, why they do this, we don't know. It seems logical. It is a way to explore space in the same way that run and tumble is for bacteria. You bias the random walk, you can climb up a gradient, etc. But it's so far eluded full quantification to understanding how they do this, what exactly is happening inside the cell to make this happen, and why they do it. It's probably related to chemotaxis.
Thank you. And the second question is, are these experiments done in the dark?
So both in steady illumination or in dark illumination using red light illumination, if you want to avoid any phototactic response. They're very weakly responsive in the red, so we often do the experiments under red light illumination. So, yes.
Thank you very much.
Okay, let's.
One more?
Sorry.
No, not me.
Oh, I see.
Larry has a question. And Ruben has a question.
Okay, last question from Larry, because otherwise we'll be too late.
I had a feeling at the beginning of the talk that maybe you would come back to Volvox. How does this compare to the cilia of Volvox?
So, first thing to say is that some years ago we discovered that actually Volvox has metachronal waves on it. So, this is an interesting point. Chlamydomonas tends to synchronize. You put two flagella together, they tend to synchronize. You put a whole bunch of them together in this geometry with a no-slip surface underneath, and you get these metachronal waves. And to this day, I don't think the community has a decent answer as to why. But there's something about beating dynamics in the presence of a no-slip wall that produces this phase shift. But you get beautiful metachronal waves over the surface. So they're synchronized, but with a phase shift from one to the next. So that's the quick answer to the difference between Chlamydomonas and Volvox. Some organisms that are in between, most of them haven't been studied quantitatively except gonium, which I'll tell you about tomorrow, and the next one, where it seems to be quite synchronized. So there's also an issue of getting to a large enough scale, because gonium only has 16 cells, a large enough scale at which you start to see this metachronicity. So that's another, piece of evidence for what might ultimately be a theory for why you see that. Does that make sense?
How many metronomes have you tried to synchronize?
I've only gone up to 5. Yes, but there is a video on YouTube which I think, unless it's AI, has hundreds.
Do they get waves or no? They synchronize?
I don't think I've seen waves. You can see transient waves as it's getting there, but it tends ultimately to synchronize.
Yeah, I'm looking at the simplified basal structural line. It's, given a certain number of nodes, there are many ways you can connect them. Is there any reason for one might be preferable to another, sort of from the...
So there's a very famous article which I commend to you. In fact, I'm going to put a little bit of a reading list online for you. And the article is entitled, "Nothing in Biology Makes Sense Except in the Light of Evolution." It was written many years ago. It's a fantastic article. And I think you have to think of it that way.
Oh, that's the question. Right.
Yeah, so what is the evolutionary advantage of this? Is it escaping predation? Is it coordinating motion toward the light? So somewhere I believe it's there, but you can't just think of it as an engineer, I would design the system this way. You have to think that it evolved. And I don't know, I always use JFK Airport as a good example. You wouldn't design it that way from the beginning, but it evolved and now you're stuck with it. And so this is, I think, the proper answer.
Okay, let's thank Ray again.